thank you for the invitation to join you this morning. It's wonderful to be here. I grew up not too far from here, so I always consider myself a New Yorker in my heart. So I'm here today. I come down from Boston. At Dana-Farber, I'm going to tell you about a new department and a new type of technology that's been emerging over the past several years. As we know, the global burden of cancer is enormous. Probably every person in this room has a personal experience with either their own health or with someone they love experiencing cancer. Dana-Farber's mission has always been to research, to treat, and provide the highest level of care. And there's been tremendous progress, but yet we see that the burden of cancer on the entire world remains enormous. So I actually started at Dana-Farber in 2001. I was a clinical research coordinator. I had been moved growing up around here when the movies used to start. I don't know if anybody remembers this, but they would show a video of the Jimmy Fund, which is our pediatric oncology program. And they'd pass around a canister, and I was so moved by this that my senior year of high school, I said I was going to be a pediatric oncologist at Dana-Farber. So not pediatrics, but here I stand. And Dana-Farber has been such an important part of the growth and development of cancer drugs in the United States and worldwide. And our mission has always been to harness the research powers of Harvard Medical School, of the academic community through partnerships nationally and internationally to grow and to develop the best cancer therapeutics and do the best possible research that we can to understand the fundamental biology of cancer to move this field forward. And so these are just a few statistics about the Dana-Farber Cancer Institute and some of the influence. And I think one of the things that's really important to underscore is that Dana-Farber has been involved in over 50% of the drug approvals through clinical trials of the available FDA-approved drugs for cancer therapy. And so these are some of the historic innovations that we've seen. Some noteworthy things are the definition of Li-Fraumeni syndrome, and again, many of the novel therapeutics that we see and use worldwide. And so what I'm going to talk about today, and I'm going to piggyback off the comments just made about how we have to think different. So when I began over 20 years ago, I was assigned a drug, STI 571. This was a novel new therapy. It was our first targeted therapy. Historically, we've really just been using chemotherapies that targeted the cell cycle, that attack things that were rapidly growing. And this drug was being used in prostate cancer. Well, probably not the right application of that drug, but that drug is now known as Gleevec. And that really ushered in what we now see and benefit from in terms of our targeted therapies, our immunotherapies, and a revolution that's really occurred over the past 2 decades. But I would argue that this approach has always been reactive. We have targeted cancers that are already developed with the therapies, and we know that our best chance of curing patients is when we find them early. So what I'm going to talk to you today is about how we move the field forward, how we think differently about how we approach cancer, and what resources we put towards the early detection and interception of cancer. What patients want and what the healthcare system needs is cure. So how do we get to cure? And so as you see here, if we find cancers when they're either in the precancerous stage, many cancers have precancerous stages, or localized, that is when we have the best chance of cure, particularly for some of our most lethal cancers, like pancreatic cancer and ovarian cancer. This is a demonstration of the survival rates of colorectal cancer. Again, when you find a cancer when it's localized, stage 1 or stage 2, it's over 90% curable. But once it has the opportunity to spread and extend and grow and metastasize, survival and prognosis are much poorer. And so how do you disrupt the cancer model, so to speak? How do you take on that ostentatious goal? Well, I think it's multifactorial. First and foremost, where we do have pre-malignant conditions, can we identify them? And can we start to study therapeutic interception before things become a cancer? Number 2, how do we find novel techniques? How do we find these cancers when they're earlier? We're going to talk about the current screening that exists and opportunities to enhance this. And finally, how do we partner this with research? As was just said, we need clinical research. The field, it takes time, and the field often moves more slowly than we'd like. But how do we harness the potential of commercial innovation paired with strong academic research? And so in the past year, Dana-Farber started a new department. It's called the Centers for Early Detection and Interception. And while it may sound very intuitive that one focus on this, that is not what has historically been done. Again, most of the medicine we practice is reactive rather than proactive. And what this does is it says we know that there are populations out there that are higher risk. How do we identify those populations? And how do we enhance screening? How do we potentially intercept early? And how do we optimize the care of people who may be at a greater risk? So this includes patients who have hereditary factors such as genetic germline mutations, the BRCA mutation, Lynch syndrome, exposed high risk. Our cancer survivors are at extremely high risk of secondary malignancies, both potentially from genetic predispositions, but also from having received cancer therapeutics. But there are other lifestyle issues that expose people to higher risk, whether it be through smoking history. Obesity is now the second leading cause of cancer worldwide. Some people have precursor conditions, as I mentioned, and then we are also very engaged in the process of the development of multi-cancer early detection tests. And so, historically, all patients that come to Dana-Farber have a cancer diagnosis. What we've now ushered in is a new era where we are seeing patients before they have cancer. So as you see here on the right, there is a wheel that shows the array of programs that we now offer. My background as a medical oncologist is in an incurable blood cancer called multiple myeloma. It has a very defined precursor condition, monoclonal gammopathy of undetermined significance and smoldering myeloma. So we have one of the preeminent programs for precursor disease where we study the basic biology of disease evolution, and then when patients are at a high risk of evolution into symptomatic cancer, we offer clinical trials. We've taken the work that we've done over the past 5 to 10 years in this disease model and replicated that across other cancers that have precursor conditions. In addition, so we have this within the hematologic malignancies. We've started programs in gastroenterology for Barrett's esophagus, for IPMNs, for high-risk patients who have BRCA mutations. We now offer advanced gynecologic screening. For patients who have oral precursor lesions, we have a head and neck clinic. And finally, what I'm going to spend the rest of this talk talking about is my own multi-cancer early detection clinic. And so when we think about the higher level, what a patient journey would look like, in a novel world of medical care where we're not reacting, we think about risk assessment. What are a patient's individual risk factors? And we heard that personalized medicine is here. It is. So how do we define what someone's risk factors are? Going beyond germline mutations, incorporating lifestyle influences, and factors that may ultimately determine a patient's long-term risk of developing cancers. How do we screen patients in a novel fashion? We'll talk about the fact that right now we only screen for 4 cancers, and that over 70% of cancers do not have existing screening tests. So how are we a part of the science that moves screening forward? How do we intercept? What are the best ways? We've learned through the examples of prostate cancer that not everybody we find with prostate cancer needs to be treated. So how do we identify the patients who properly need to be intervened upon, and what are the right interventions? And how do we do this in a comprehensive way so that we're considering a whole patient and integrating their care potentially within multiple centers? And so the foundation of Dana-Farber's success has always been the bench-to-bedside approach. So how do we work within an academic community? How do we collaborate nationally and internationally to bring the best science forward? How do we conduct those clinical trials so that new therapeutics or new screening tests are properly vetted? And how do we bring them to patients, and how do we bring them to all patients, not just the people who are fortunate enough to live in large cities and have access to care at tertiary medical centers? And so I will now begin speaking about multi-cancer early detection. So before you, we see the types of screening tests that are available and recommended worldwide. In the United States, it is recommended that one be screened for colorectal cancer, breast cancer, and cervical cancer. If you have a significant smoking history, then lung cancer screening is recommended. And that is the extent to which we currently screen for cancer. But we know that 70% of cancers in the United States and 50% internationally of cancers do not have current screening methods. Again, reinstating what I had said before, we know that cancers are most curable when they're found early. What you see before you are examples of both pancreatic and ovarian cancer, which are two of our most lethal cancers in the United States. And our best chance of curing these is when they're found early. So why is it that we don't screen for all cancers all the time? The problem is there are too many, and the incidence is not high enough that you could find enough cancers to warrant the amount of effort that would have to go into screening. It would be an unreasonable thing to ask of a primary care physician to screen for 50 different types of cancers. So if we aggregate different types of cancers into singular subsets or small batches of screening tests, then potentially we have a new way in which we might be able to identify and better screen for more sporadic cancers. And so one of the new fields that has emerged in the last couple of decades is the idea of liquid biopsy. And it's not just blood-based, it can be urine-based, breath-based even. These are sciences that didn't exist 20 years ago. About 20 years ago is when we first sequenced the human genome. And since that time, we've gone on to sequence the genomes of different types of cancers and create libraries from which we can then look within the blood, in this example given, find fragments of DNA, and look at them, compare them to what is normal versus what is abnormal, and potentially then identify a type of cancer from within someone's blood. And why is this attractive? Well, one of the things that matters most to me is the idea that we can improve the healthcare system. Many people in this room experienced trying to practice medicine through a pandemic, and it pushed our healthcare system nearly to the brink. So it's good to think about how we have better therapies or better screening options. We also have to think about what is the implication of these new screening options on an already burdened healthcare system. And I would argue that blood-based screening has the potential to mitigate some of the burdens that we currently have as a complement to existing screening. In addition, it has the potential to expand access. There are current tests available. There's a singular clinically available test that is not FDA approved that's quite costly. So how are we conscious of not exacerbating disparities in our goal of alleviating them? One of the things that I think is most exciting is just the potential to not only expand the type of cancers we can screen for, but find them earlier. So I think that we are at really the beginning of what has a potential to be as revolutionary as the therapeutic revolution we've seen over the past 2 decades. So we opened a clinic at Dana-Farber focused on really this idea of how do we bring good tests forward, how do we study them rigorously in the same way we would as drug. If we took the same intellectual power that we use for how we study and approve therapeutics and applied it to screening and to early detection and early interception, what can we accomplish in the next 2 decades? And so our clinic right now, in the tests that have been done, for the GRAIL Gallery test, which is the single clinically approved test, Dana-Farber was part of the team that led the Pathfinder study that led to the approval. And when this test is used in the general population, only about 1% of those screened will have a positive test. And again, these are screening tests, they are not diagnostic tests. So a diagnostic process needs to ensue to adjudicate the result. And so we do that at Dana-Farber. The positive predictive value of the existing test is only 43%. So there's a responsibility as you introduce new methods to take care of the patient who may not actually have a cancer as you conduct this workup. So when a patient does have a positive test and is found to have cancer, those patients are ushered into the appropriate clinic. But when they do not, I follow them clinically. We do blood banking. We take extensive lifestyle histories, and we follow patients accordingly. And so a typical visit for a program like this includes a visit with an MD, genetic counseling. So Dana-Farber has 27 genetics counselors. For every patient who comes into our clinic, they're screened for the need to see a genetics counselor and offer germline testing if appropriate. We do a thorough diagnostic workup, and as I mentioned, we either follow those patients ongoing or refer them to the appropriate oncology clinic. We've also developed several other programs in this Centers for Early Detection and Interception, which I think I've mentioned, but are also worth highlighting. This includes our early detection and interception of GI tumors. There are a number of benign tumors that currently do not have homes. lymphomas. We see things like IPMNs, or we see things like Barrett's esophagus very commonly in our clinical practice. But how do we learn from those patients who are at highest malignant potential, and how do we potentially intervene? Same thing is true for our genetics program. Dr. Kevin Elias has been heavily involved in our germline populations and in the development of ovarian cancer screening tests. How do we promote this type of endeavor? How do we support the type of work that's being done to find rarer cancers earlier? And one of the most important things is how we collect data. So we've developed the In Advance study, and I think the future of oncology is moving away from centralized IRBs, centralized research, and trying to go to decentralized approaches that enable web-based access or potentially global access. And so this is our banking study where we are collecting blood, stool, urine on all individuals who come to Dana-Farber, as well as a very extensive medical and personal history, with the goal of trying to catalog and understand better patients who are at highest risk for developing cancer. As I mentioned, this is a web-based platform, and so these studies can be done in places such as Quest within the United States. It does not require a trip to Boston to participate in this type of research. And when we think about the myriad of programs, this is just kind of a peacock view of the different programs that have grown. We started with just a handful, and in the year's time since this program opened, you can see the development of programs that have sprouted from it, and these are the providers. And what I would say to you is when you build it, the people come. So when you create a program and set a foundation for this type of thinking, people start to come. It's not just the patients, it's the physicians, it's the researchers. So having the infrastructure and creating a pathway forward is necessary for this type of innovation. As I mentioned before, I'm a blood cancer specialist, and we have a long-standing program in plasma cell dyscrasias, which has now been extended to the leukemia and lymphoma precursor spaces. And really there are three prime aims of our program. One is to screen early. In the case of multiple myeloma, we know that there are increased incidence in patients of African American descent. We launched the largest international study of screening patients, P-Crowd study, which is offered in multiple different countries, including South Africa and Israel, as well as the PROMISE study screening individuals with family history and of African American descent. A key piece of this that I mentioned earlier is it's not just sufficient to find someone early, it's to understanding their risk. So how do we do the basic science? I work very closely with Dr. Irene Goldriel, who really studies the biology of disease evolution. So if we can better understand that, then we can better risk stratify, and we can target the patients who will benefit most from intervention. And finally, early interception. We offer clinical trials of some of our most novel drugs, CAR T-cell, bispecific T-cell engagers in smoldering myeloma and highest risk. And this is really a unique paradigm for interception of premalignant conditions. These are some of our disease-centered champions. This is our website. If one is interested in contacting the Centers for Early Detection and Interception, if someone is interested programmatically in talking about developing a program, I'd be happy to speak with you. And I want to end with a thought that the light bulb was not the result of incremental improvement of the candle. As was said earlier, if we really want to illuminate the world and make progress, then we do need to think different. Thank you very much.